Hello guys, welcome back to one more video session. In this session, we are going to talk about the mechanism of transcription in prokaryotes as well as in eukaryotes. In the initial stages of the transcription, both in prokaryotes as well as eukaryotes are same. The synthesis of RNA mechanism is same. Exceptions are there like the RNA polymerase. In the case of this, there is only one RNA polymerase is enough. In this case, there are different types of RNA polymerases are there. We'll talk about it. Once the RNA is synthesized, in eukaryotes, it requires post-transcriptional modification. I'll tell you why. Okay, so it is going to be lengthy, whereas here this is very short, very easy to learn. So first we will talk about the prokaryotic transcription. First we will draw the DNA strand. So we will mention its ends also, 3 prime, 5 prime, and this is the antiparallel strand. So we just write 5 prime, 3 prime like that, and even we put the nitrogenous bases like this, and not required. Okay, so like this, done. It's very easy now. We know this site is called as the promoter, so we just write P for promoter, and this is the terminator. Got it? Okay. See here, the promoter has got certain base pairs, even though we cannot call it as gene because this is the gene. This is the structural gene. And here in the structural gene, you need see two types of strands. One strand which actually produces the RNA, which gives or shares its information to produce the RNA, that is called as the template strand. We call it as template strand, and this one is coding strand. The name is strange, even I told you why coding strand. It will not code for any RNA. Okay, so we will talk about the promoter first. In the case of this promoter, so let me just put a box. You know about it. I have explained about a transcriptional or transcription unit in the previous video session. Fine. So here, let me just name it. See, this is the coding strand, that is non-template strand. Even I can mention this as non-template strand, correct? This is the template strand, and in the non-template strand, you could read some sequences. Okay, so we write here. See, I'm going to write these sequences here. Okay, that is within the non-template strand of the promoter. These sequences normally about 40 to 60 base pair sequences within the promoter. Let me just name some sequences. I cannot write till 60 base pairs. Okay, some important base pairs, that is T, T, G, A, C, A present within the coding strand or non-template strand. This side, I'm talking about this side, not the template strand. Okay, and again here within the promoter you could see another sequences called as it is read as TATATAAT, tatart, like that. Okay, this is very important. So now, which is present here, the same sequences present within the promoter. The first step of transcription is initiation. For the initiation, what is required is RNA polymerase. DNA dependent RNA polymerase is very important. Okay, so we just draw the RNA polymerase. How many polypeptides are present in RNA polymerase? 6. 2 alpha, 2 beta, 1 omega and 1 sigma factor. So sigma factor is very important. So I am going to just write sigma factor within the RNA polymerase. I will not mention the other remaining 5 polypeptides of the RNA polymerase. So let us name it RNA polymerase. Only one RNA polymerase is enough for the process of transcription. Okay, in prokaryotes, that is enough. So now the sigma factor first actually identifies this particular sequence TTGACA. Note this point, TTGACA first, that is present on the coding strand or non-template strand of the DNA. Okay, so this will be recognized by the sigma factor of the RNA polymerase. So it's going to bind to this sequence. So that is enough for the remaining 5 polypeptide chain of the RNA polymerase to bind to the promoter. So for this, so for example, if I remove only one polypeptide chain, that is sigma factor is already attached to this, what is remaining? 5 polypeptides. So that will make up the core RNA polymerase. So I'm going to write core here. So remaining 5 polypeptides, that is core RNA polymerase enzyme, I will write sigma also here. So that's going to bind to this box, that is TATAAT sequence. Note this point. This sequence, particularly called as the sequence which reads TATAAT, is called as Pribnow box. Note this point, very important for your competitive examination. Pribnow box is the sequence which is present only within the promoter region of the DNA, which is normally present on the non-template strand, not on the template strand, okay, which helps in the recognition of DNA by the RNA polymerase. That's it. Got it? So this is the thing. So RNA polymerase now binds to the promoter. Done. So now that is called as initiation. So when the core RNA polymerase binds to this Pribnow box of the prokaryotic DNA, and this mechanism will allow the RNA polymerase to transcribe. That is, it allows RNA polymerase to move in this direction. So got it? So this is RNA polymerase. Now it's going to bind here. So for example, this is now RNA polymerase. It's going to bind to the promoter. First, sigma factor recognizes these 6 base pair sequences present within the promoter. Then the RNA core, that is RNA polymerase core, will recognize the Pribnow box which is present within the promoter. So everything is now attached. The entire R6 polypeptide chain of the RNA polymerase is now already bound to this promoter. Now it starts moving. When it moves, there are 2 things happens. That is, hydrogen bonds between the template and the coding strand separate. Got it? Separate. Why it separates? That's what, like, here you need to understand this. I have written TATAAT. There is no G and C. Note this point, correct? There is no G and C. If guanine and cytosine is present, then obviously there is 3 hydrogen bonds between them. So to break the hydrogen bonds, it requires a lot of energy. So this is the recognition side. Only this is containing A and T. So we can also mention it as AT rich region is required for the recognition of DNA by the RNA polymerase, so that it can break. From here onwards, the breaking starts. So whatever the nitrogenous bases are there, right? They are linked by hydrogen bonds. So we need to break this. So the process of allowing the RNA polymerase to move within the structural gene. This is now structural gene. From here till here, structural gene. So that is called as transcription clearance. Transcription clearance. Remember it, for the transcription clearance we need Pribnow box, that is AT rich region, so that it can easily break the hydrogen bonds, because adenine and thymine 2 hydrogen bonds, right? It's very easy to break the hydrogen bonds. Done. Got it? So that's it. So that is the initiation. Okay, so for the RNA polymerase to work, they need the energy, that is nothing but the cofactor required for them to work, that is magnesium. I think I have mentioned it in my previous session, I don't remember it. Okay. So first step is done. We'll go to the second step. Got it? So now we mentioned, I use a red color here. So RNA polymerase is already attached. Initiation is done. Next is elongation. So elongation is nothing but it is the process of production of RNA. It could be rRNA, tRNA or mRNA, doesn't matter, from the template strand of the DNA by the RNA polymerase. DNA dependent RNA polymerase enzyme is nothing but elongation. So in elongation, what you see is breakage of H bonds between the template as well as non-template strand of the DNA, and then the movement of RNA polymerase towards the terminator region. That is actually the thing you see. Got it? And another important mechanism is when the hydrogen bonds between the 2 strands of the DNA is broken. So what's going to happen is, when they are broken, so you will see a bubble shaped structure, like the DNA, both DNA now separated. Got it? So both now separate. So now I am going to, anyhow it's broken now. So what we are going to do is, we just draw like this. Matlab, you could see the DNA strands are now separated. Correct? And you could see a bubble shaped structure, bubble. Okay, so this bubble is seen only in elongation, that is done by RNA polymerase enzyme. So this is called as transcription bubble. Remember it. Transcription bubble is formed by the RNA polymerase during elongation process. Okay, now in the elongation process, what's going to happen? The RNA polymerase, right here, in the elongation process, the RNA polymerase will bring ribonucleotides. See, I'm telling you that it will bring the ribonucleotides, not deoxyribonucleotides, because it's going to synthesize RNA. So ribonucleotides, that is adenine rich, guanine, cytosine and uracil ribonucleotides. Okay, so now what's going to happen here? Let us say now here the template strand is there. Now here the RNA polymerase is starting to move towards the terminator region. So it's going to bring the ribonucleotides. So we use a blue color here. So ribonucleotides will be brought. How exactly? See, for example, here the RNA polymerase is now moving and the ribonucleotides are brought towards the 3' end of the template strand like this. But for the RNA, it is 5', correct? Because it's moving towards this direction. So for RNA, it should be complementary. Okay, so like this. So now, so this is 3', sorry, not 5', because this is 3 for the DNA template strand. And obviously, if the RNA is going to be formed from like this, so this end of the RNA is 3' opposite, and this end should be 5'. That's it. So now, the growing ribonucleotide chain, that is mainly because of RNA polymerase activity, is going to add the ribonucleotides in the 3' end. Remember this, very important. Got it? So for example, if this is the DNA, let me just 5' and 3'. So I will not write about draw the template strand, it will confuse you. So this is actually the ribonucleotide which is now added by the RNA polymerase towards the 3' end. So one is added, next is now brought, which is now linked. See, the next is linked. So as a result, what's going to happen? The first ribonucleotide is going to shift towards this direction. So see, got it? So you see a long chain because the RNA polymerase is also running. Are you getting my point? This is RNA polymerase. So when it runs, okay, so this is enough. Promoter region and this is the terminator. So now the RNA polymerase is already attached. So it's going to move now like this. So when it moves, it's going to bring the ribonucleotides. So first ribonucleotide is added, second, third. So what's going to happen? This is actually the thing. What you're going to see, sorry. Okay, so I think it will confuse you. So this is the first ribonucleotides. So now the RNA polymerase is shifting. So first will attach to the RNA polymerase itself. Next is the second ribonucleotide. See, next is the third. Got it? So the yellow color one, the first ribonucleotides, when it reaches the terminator site, the RNA polymerase, this will remain as it is. Are you getting my point? So this is the thing. So this will be 3' end and this will be 5' end. So that means for the growing RNA, the ribonucleotides are added towards the 3' end, not towards the 5' end. The 5' end, whatever the ribonucleotide you see here, this is the ribonucleotide towards the 5' end. This is the new ribonucleotide added. This is the first nucleotide added. Are you getting my point? So finally, when that is all about the elongation process. Hope you noted this point. Very important. Got it? So replication will be always in the direction 5' to 3'. Now got it? So 5' to 3'. This is the thing, very important. It's very easy. See, 3' to 5'. So the opposite, that is RNA itself, that should be opposite strand, correct? 5' to 3'. Done. Then once elongation is done, the last step is termination. What is this termination? Termination is nothing but when it reaches the terminator sequence, there is a protein called as the Rho factor or Rho protein. Remember this Rho factor, and even I can also write this as Rho protein. This Rho protein will detach the RNA polymerase. It will tell the RNA polymerase, okay, enough, your work is done, okay, leave, done. When the RNA polymerase is detached, along with it there is a long chain. What is that long chain? The long chain is nothing but this. This is now the RNA. So RNA is attached. Got it? So this side, that is actually the side which is attached to the ribonucleotide which is attached to the RNA polymerase is 3', and this side is new ribonucleotide is there, that is 5'. So 5' to 3' is actually the initiation of what you call this one, RNA transcription or production of RNA through transcription process. That's it. So this is the thing. Got it? So finally both will detach when they reach the terminator sequence where rho factor is there. Got it? So rho factor is there. There is one more hypothesis that along with the rho factor there is a sequence which is here, which is called as repeated adenine sequences. Continuous A will be there, poly A will be there, many A's will be there, okay, in the template strand. So that means the RNA which is now produced will add U, correct? Yes, right? U, that is repeated uracil sequences on the RNA or mRNA. So this makes them uncomfortable, which, see, many U's are produced, so we can also call it as hairpin. So this hairpin is produced within the RNA, new RNA. So that makes it uncomfortable so that they detach. There are two factors. One is rho factor, the other one is many A's will be there within the terminator that will detach the RNA polymerase. Done. So that RNA also gets detached. These are the things actually. Okay. So that's all about the prokaryotic transcription. So here I draw only one diagram. You need to draw three diagrams. So first one is initiation, that is normal diagram. Next is elongation, so you need to draw this bubble. Then termination means, okay, termination, how do you draw the diagram? So this is the polymerase enzyme. Okay, so write rho factor here, somewhere else here, and just draw a long chain of RNA. So here 3 prime and this is 5 prime. Done. Got it? So that's all about it. So you need to detach the RNA polymerase and this RNA from the DNA, that is from the terminator region. That's it. That is actually termination. So please remember it. You need to draw 3 diagrams for your board examination. Here I did not draw it. Okay, within one diagram I explained everything. So that's all about in the case of prokaryotes. Done. Now the interesting factor is when normally for your competitive examination, when this RNA polymerase enzyme is running towards the terminator, obviously we know that this mRNA is synthesized continuously. So 5 prime to 3 prime will be like synthesized. During this process, these prime like the mRNA which now comes out of it, some portion of the mRNA is coming out of the DNA, that is detected by the ribosome. Ribosomes will be there because everything is happening within the cytoplasm of the prokaryote or bacteria. So ribosome comes and now they just bind to it. Done. So that means transcription and this is translation happening at the same time simultaneously. When a small portion of the mRNA is synthesized during transcription process in the bacteria, at the same time the ribosome also comes and bind to the mRNA. Done. So that the message is now translated to produce a protein. So that is simultaneously. So that means in the case of prokaryotes, transcription and translation occur simultaneously, that is within the cytoplasm, and the rate will be very fast. So within a fraction of a second proteins will be synthesized. Why? Because if you look into prokaryote, the cell division, that is production of new daughter cells or new bacteria, will be within 20 minutes. This is very important. So transcription and translation requires only 18 minutes. Only 18 minutes in the case of prokaryotes. Done. So that's all about the prokaryotes. Now we have to talk about the eukaryotes. Now we will discuss about the transcription in eukaryotes. There is no difference, like much difference between the prokaryotic transcription and eukaryotic transcription. Both are same, and even the direction of production of RNA is also same. That is 5 prime to 3 prime. Same. Everything is same. Everything is same. But here I have mentioned it that RNA polymerase with the help of magnesium. How many RNA polymerases are there? Only one. Only one is enough to synthesize any type of RNA within the bacteria. So here in the case of eukaryotes, there is a clear-cut division of labor. That is, for the synthesis of a specific RNA, they need specific RNA polymerase enzyme. So first we are going to write that is the division of labor. Please note this point. So division of labor in the RNA polymerase in the transcription of eukaryotes. The first type is RNA polymerase. Let me just write RP. RNA polymerase 1. Roman number 1, then RNA polymerase 2, then RNA polymerase 3. There are 3 different types of RNA polymerases only seen in eukaryotic transcription or eukaryotic DNA. Here RNA polymerase mainly, it is essential for the synthesis of a type of RNA called as rRNA. So that means you may get a question, rRNAs are synthesized in eukaryotes by RNA polymerase 1. Okay, not only rRNA, it also particularly in rRNA there are 3 types. Let me just write. The first type is 28S type rRNA. Another one is 18S type of rRNA. The other one is 5.8S type of rRNA. Please note this point. Very important. The function of RNA polymerase 1 is to synthesize ribosomal RNA. There are 3 different types of ribosomal RNA. That is 28S, 18S, 5.8S. Please note this point. Very, very important. Very important. Then, for RNA polymerase 2, in eukaryotic transcription, it is going to synthesize a type of RNA called as hnRNA. So, what is the meaning of hnRNA? Hn means heterogeneous. What is the meaning of hetero? Hetero means different. I will tell you why. Okay, heterogeneous nuclear RNA. Remember, that is by RNA polymerase 2. In this session, I am going to talk about RNA polymerase 2's function. How exactly they help in the transcription. So, when you are writing in the annual examination, if the question is related to eukaryotic transcription, please understand this. Don't write RNA polymerase. Write RNA polymerase 2. Mention it. That's it. That is the only modification you need it. In this case, only RNA polymerase you are writing. In this case, you are writing RNA polymerase 2. Okay. So, that's going to produce hnRNA. You are going to write hnRNA synthesized. Instead of RNA, you are writing hnRNA. That's it. The modification is required. RNA polymerase 3. Actually, it synthesizes tRNA, not only tRNA, it also synthesizes 5S rRNA, the second one. The next is snRNA. SN means small nuclear RNA. See, note this one. Learn everything. Okay, it's not that difficult, it's very easy. RNA polymerase 1, what is the function? RNA polymerase 2, what is the function? RNA polymerase 3, what is the function? See, tRNA, 5S rRNA, small nuclear RNA. You got it? That's what I said in my previous session. There are millions of RNAs. So, in the eukaryotes, there is a clear-cut division of labor. That means each RNA polymerase is specific for a synthesis of specific RNA. Done. So, this is the thing. In this session, I am going to focus mainly on this RNA polymerase 2. So, let me just erase this because I need space. Okay. Now, I have mentioned that in the promoter sequence, there is a particular, in the promoter region, there is a particular sequence called as box. In this case, you see the same sequence which helps in the recognition of RNA polymerase 2 in the promoter region. So, what I am going to do is simple, I am not going to explain the concept, but this is the promoter. So, just mention. Okay, so the promoter region. In the promoter region, particularly in the non-template strand or coding strand of the DNA, there is a particular 6 base pair sequences. So, let me just write the 6 base pair sequences. A. How many are there? 6 base pairs are there. In the case of prokaryotes, instead of A, T is there. That is called as box. In this case, this box is called as the Tata box. Again, it is AT-rich region that is helpful in the breaking of the 2 strands of the DNA, correct? By the RNA polymerase enzyme with the help of energy also. So, remember it, Tata box is seen only in eukaryotes, whereas box is seen only in prokaryotes. This is the Tata box seen in the promoter region of the particularly which strand, that is non-template strand. Okay, the other name of this is also called as Goldberg-Hogness box. Remember this, Goldberg-Hogness box is the another name of Tata box. That's it. So, in the examination, for board examination, no need of mentioning all these things because it's very easy. You just write what's going to happen. Okay, RNA polymerase first binds to this promoter with the help of the sigma factor, done, that is initiation, then elongation, the hydrogen bonds are separated between the 2 strands of the DNA, then RNA polymerase now runs. Got it? So, when it runs, it's going to bring the ribonucleotides in the direction that is, it is going to add the ribonucleotide towards the 3' end. So, synthesis takes place from 5' to 3' like that. You just add it. When it reaches the terminator, there is a factor called as rho factor, that is nothing but rho protein, which detaches RNA polymerase and the RNA. Done. So, that's all about it actually, the transcription. For neat only I am telling everything. Remember, Tata box, that is Goldberg-Hogness box like that. Done. And terminator same. Okay, you see poly A region that is having more adenine residues will be seen. Okay, that is nitrogenous synthesis. So that's all about it now. Now I am going to talk about post-transcriptional modification. Okay. So here you need to listen to me carefully because it leads to confusion, not difficult, leads to confusion. I am going to write a word called as cistron. So what is this cistron? And again I am going to write the types of cistron. There are two types, polycistronic and the other type is monocistronic. Okay, so what is this cistron? Cistron is nothing but gene. It is the segment of DNA which codes for a particular protein, that is gene. The other name of the gene is cistron. This is very specific in the molecular biology. You got it? This term is very specific because gene means like you can include promoter, you can include terminator of a DNA segment, even structural gene, everything, that is gene. But which is exactly gene? That means like the promoter sequence is also there, terminator sequence is also there. They are not going to help in the synthesis of protein. Okay, they are help in recognition and termination of the transcription. So cistron is nothing but structural gene which now actually produces only the protein or amino acid sequences in the ribosome. So that is cistron. So what is the definition of gene now? It is the functional unit of inheritance, that is gene. Cistron is the segment of DNA which codes for a particular protein or particular polypeptide chain. Correct? That is cistron. There are two types. One is polycistronic, that is seen only in prokaryotes. Very simple, very easy. Prignobox starts with P, polycistron P, prokaryote P, no confusion. Polycistronic means this particular gene is there, right? Now I can call it as cistron or particularly polycistron in the case of prokaryotes. This one mRNA which is now produced due to transcription process, it's going to produce more than 3 proteins, different proteins. So this one mRNA can produce more than 3 proteins. If multiple proteins are coded by a single gene, then that kind of gene is called as polycistronic. So that is seen only in prokaryotes. Can you tell me why? In the case of eukaryotes, that is monocistronic. So now you understood what is mono. Mono means single. So single protein will be synthesized from a single cistron or from a single gene. In the case of eukaryotes, prokaryotes, multiple proteins from the single mRNA or from the single gene. Got it? So why? Very simple. It is single-celled organism, the prokaryote. It requires enzymes and the length of the DNA is also very short. So it is a modification, evolution. So because of that modification, one DNA segment, that is the gene or cistron, will produce one mRNA. That one mRNA is going to produce different types of proteins. You are going to understand it in lac operon concept, that is about prokaryotes. In this case, it is monocistronic, that is single protein will be synthesized from single gene. Remember it? Why? Because our DNA's length is very long compared to the prokaryotic DNA. We need a specific type of protein. A single gene cannot synthesize different types of proteins, multiple proteins at the same time. It may lead to complications, correct? So this is the thing. So to avoid the complication, because you know our organization is complex compared to the organization of the bacteria. Done? Got it? Okay. So this is the thing. Now again, in the polycistronic, whatever the mRNA which is now synthesized from transcription process, I will just draw it. So this is the mRNA, 5 prime to 3 prime. So this is the mRNA and it has got the sequences that is coded by the template strand of the DNA, correct? We know it. Okay. And in this case, whatever you see here is nothing but useful bases, so that this can produce a type of protein. See, some base pair will produce a protein and this base pair will produce a protein. So this base pair will produce a protein. So like that, you see multiple proteins and all the base pairs are now useful. Got it? Useful. So that's what I said, like polycistronic obviously like produces mRNA, mRNA will produce different types of protein. So there is no complication. But when it comes to eukaryotic cistron, that is monocistron, you see complication. Please note one point. Eukaryotic genes are called as split genes. Very, very important for both your board as well as competitive examination. Why eukaryotic genes are called as split genes? What is the meaning of split? There is some complication should be there on the produced mRNA. That split gene. Split gene is mainly seen in monocistronic, that is eukaryotic. gene, eukaryotic gene. Okay, so fine. Yeah, let me just explain what is the split gene. Split gene means the mRNA which is now synthesized at the end of a transcription process, that is 5 prime to 3 prime. Here, the sequences which are now complementary to the template strand, obviously. So I'm just highlighting this. See here, highlighting this, which now contains some regions that mRNA contains blue color, blue, yellow segments, which is now coded by the template DNA, which contains some useful sequences, that means it's going to produce the protein, and some useless sequences. That means in the monocytonic gene, if you look into the segment of the DNA which now codes for a particular protein or produces particular RNA, some portion will be within the structural gene, some bases will be useful, some bases will be useless. Got it? So we need to remove that useless portion of the RNA, otherwise the protein will be kind of like abnormal. So to avoid it, so here this portion, yellow color one, is now called as introns. I'll tell you what is the meaning of introns. The blue color portion is called as exons. Very easy. The RNA contains exons, introns in eukaryotes of monocytonic gene. Okay, so as a result, which is called as split genes. What is split gene? Eukaryotic gene or eukaryotic RNA, which is produced due to transcription process, contains exons and introns at regular interval. So that is actually called as split gene. Now I want to remove this introns. Why I have to remove it? Introns are useless, whereas exons are useful. So exons are the sequences which is helpful in the production of proteins. Actually, exons means expressed sequences. Note this point: expressed sequences on the mRNA, on the synthesized RNA. Expressed sequence, EE. Very easy, no confusion, right? EE. Exons, E is the expressed sequences, which are actually used for the synthesis of proteins in the ribosome. Whereas introns are non-expressed sequences. Non-expressed sequences. Any confusions? No, right? So for example, let me just erase this. Hope you understood what is split gene. Split gene is nothing but a type of monocytonic gene which contains exons and introns at regular interval on the processed or the produced mRNA. Okay, so here this is now, let me just write the heading also for it. Okay, post transcriptional modification. And when I was discussing about the division of labor of RNA polymerase, as I said, the RNA polymerase 2 will produce hnRNA. So this RNA cannot be called as mRNA now because it is a nascent RNA. Nascent means nascent RNA. Okay, so nascent RNA is seen only in eukaryotes, not in prokaryotes. Prokaryote matured RNA. This is nascent, is not matured RNA. Got it? It has to undergo certain modification. Now it is called as hnRNA. What is hnRNA? Heterogeneous nuclear RNA. Heterogeneous means two different things, correct? Exon, intron, exon, intron, exon, intron. I got a model here. Okay, not a model, a pipe. Okay, so this is the hnRNA which is now produced from the DNA. See, this portion is called as exons. The white color portion is intron, exon, intron, exon, intron, exon, intron. So this is split gene. Got it? So the same sequences will be present on the DNA template strand. Why? Obviously, you know that if RNA is containing this type of exons and introns, the gene, that is the structural gene, the template strand should also contain the same. Correct? So that's it. So eukaryotic genes are monocystonic but split genes, whereas prokaryotic polycystonic but not split genes. There is no exons and introns. Only exons are there. Only exons. See the difference now. This is the mRNA of the prokaryote. This is the mRNA of eukaryote. So the difference. Got it? So this is interrupted, having some interrupted sequences. In this case, there is no interrupted sequences. Only exons are there. Only express sequence. Every portion, every nuclear or like the nitrogenous bases can be used to synthesize protein, 3 or 4 proteins. But in this case, only one protein. But before that, you need to remove these portions. You need to remove the introns. The white color patches are there, right? You need to remove it. So that is post-transcriptional modification. Remember. Okay, so we just write 5 prime here and 3 prime here. Got it? So within the nucleus, once the hnRNA, nascent RNA is produced in the eukaryotic DNA, that is within the nucleus, within a fraction of second, some enzymes will come for modification. So what I'm going to do is, here I'm going to use red color. So it is understood that red color is introns, the yellow color is exons. See, this is introns, yellow color is exon, again introns, like that. What I have to do is, the enzymes should remove the introns, otherwise the protein synthesized will be having some abnormal amino acids. So I need to remove this. So how can I remove this? The process is called as post-transcriptional modification. It is also called as splicing. Remember this? Hope this is a very lengthy session. I know that I am giving detailed explanation of each and everything. So watch it again and again. Okay, understand it. Splicing. What is this splicing? Splicing is nothing but it is the process of removal of exons or introns. Introns, because I need exons, because they are express sequences which contain useful sequences for the synthesis of protein. So removal of introns from the hnRNA. Once I remove the introns from the hnRNA, I can call the RNA as mRNA. Once I remove the introns, I can call the hnRNA as mRNA. Now it is a messenger RNA. It is containing proper sequences. Let us see how exactly it will be removed. The process called as splicing. In splicing, you see two steps. Step number 1 is called as capping. Step number 2 is called as tailing. You have to write step 1, step 2. I did not write, you mentioned it. Capping is at the 5 prime end. See here, the 5 prime end of the hnRNA is type of chemical which is added, which is called as methyl guanosine triphosphate. So what is the name of the chemical? Oh, sorry, I forgot to mention. It is 7-methyl guanosine triphosphate, which is added to the 5 prime end. Methyl guanosine triphosphate, like this. Methyl guanosine triphosphate is added to the 5 prime end of the hnRNA. The process called as capping. Capping is always towards 5 prime end of the hnRNA. Got it? Who adds this? There is one enzyme. Enzyme is required for the adding 7-methyl guanosine triphosphate. That is methyl. Guanyl transferase enzyme. Please note this point. Guanyl transferase enzyme which adds 7-methyl guanosine triphosphate to the 5 prime end of the hnRNA, and the process called as capping. So this is done. Next is, I will tell you why all these things. Okay, tailing is nothing but the process of adding poly means many, adenylate residues. That is adenine containing residues. Adenylate residues. Okay, who adds it? The enzyme required for this addition of poly A adenylate residues, that is poly A residues. Poly A residues called as poly A polymerase. Okay, poly A polymerase will add polyadenylate residues towards here. That is 3 prime end of the hnRNA here. So here I am adding poly A, here I am adding methyl guanosine triphosphate. Okay, so this process is called as now capping and tailing. So this is tailing. So this is the tail of the mRNA or hnRNA. This is actually the head, that is cap, head of the hnRNA. Got it? So this is the thing. Now note this point. Now once it is added, it will attract certain enzymes which is called as snRNA. So not enzymes, I better say RNA, because RNA means it is having catalytic activity like enzymes. So better say small nuclear RNA will get attracted because of capping and tailing to this site. So we just write snRNA is added. Not only snRNA, along with that there is one more protein called as snRNP. Small nuclear RNA, along with that small nuclear ribonucleoproteins Can you write this? Not nucleotide, ribonucleoproteins. Okay, we write it. Ribonucleoproteins. Proteins will get attracted to the HNRN because of capping and tailing. So now this two components, that is snRNA and snRNP, is going to form a loop now. So it's going to form a loop. That loop is called as spliceosome. See, in the sense I don't have a thread. Okay, so I have got a thread here. So let us imagine this is now the HNRNA. So there are sequences where we'll mark it also. These are all now introns. So snRNA and snRNP is going to fold this in such a way that it's going to remove it actually in future. So like this. So you see some bubbles like this throughout the HNRNA wherever introns are there. So it will be like this. Okay. So this process conducted by snRNA and snRNP in the HNRNA to remove the introns is called as spliceosome. Yeah. Spliceosome. Okay, spliceosome. Spliceosome means removal of introns from the HNRNA. Okay, splicing means. Spliceosome means, you know what is zome. Zome is nothing but a ball shaped structure, centrosome, like that. Okay, so that is actually the thing, it's a body. Now, once it is done, once loop is formed, so it's going to remove it. So this, see here, this is now removed. The introns are being removed by the snRNA and snRNP. Done. So now the ligase enzyme comes and now attaches these exons. Okay, because you know when the introns are removed, there is a gap formed. Correct? Gap is formed. So now it's going to remove everything, remove the introns. So that's all about some post-transcriptional modification that occurs within the nucleus of the eukaryotes. That's all about it. Learn each and everything. Don't leave anything. It's very important. Okay, the question could be asked from anywhere from these concepts. So I know I have made around 3 lengthy videos, and you need to watch within this day itself. Do not keep it for the next day. Okay, so watch thoroughly, write down each and everything. When you just compare this video session with the notes given, the notes means like very short important points are given, that is only for your board examination, whereas here the detailed explanation is given for your competitive examination. So stick to this. If you are preparing for your competitive examination, stick to this. Okay, so that's all about it. Hope you understood this video session, useful also for you. The other thing I just want to tell is when I take the video session, obviously I make some mistakes, like the spelling mistakes. You might have noticed it. Okay, so instead of writing something like, for example, methyl, so I may leave L. So you might have noticed it. That happens. It is not a live session. Like there is no interaction between me and you. It's not like a live classroom session. So normally I need to record this first, then I need to edit it. That is, splicing is required for it. Okay, I need to remove the introns. Only like I need to keep exons. Okay, so it's a very lengthy procedure. I need to watch again from the beginning. It's just imagine, okay, 40 minutes I need to sit and I watch it again. If any mistakes are then I need to remove the things. Okay, I need to edit it. So it requires a lot of effort also. So you need to understand that. Okay, so don't simply fast forward the video. You know that I have repeated again and again some sentences. Only thing is you need to get used to that particular word or particular concept. That's it. Okay, if I tell it, stress again and again, obviously that will be stored in your brain. Got it? So that's all about it. Fine, chalo, see you in the next week. Take care, have some fun time. Bye.